Hello everyone, my name is Verónica I Cantarelli and I am a biologist and PhD student from Córdoba, Argentina. I am also the professional in charge of the endocrinology laboratory directed by Dr. Marina Ponzio, belonging to the Institute of Physiology of the National University of Córdoba. Today I am going to talk to you about the non-invasive sampling techniques for endocrine monitoring. Although there are several methods for measuring the concentration of steroid hormones such as glucocorticoids, estrogens, progestations, testosterone, this has been based on their determination in plasma, but the extraction of blood samples constitutes in itself a stressful procedure. An efficient alternative developed in recent years is the determination of metabolites of steroid hormones excreted in feces, urine, and deposit in feathers, hair, saliva, and corneal elements, since it avoids capture and manipulation of the animal, particularly important when dealing with wild animals or when recording behavioral data, and furthermore sample collection can be performed during long periods of time. The steroids of interest are extracted from the matrix using methanol, ethanol, or an ether, and the resulting supernatant is assayed using polyclonal antibodies in an enzyme immunoassay. How it works. For the correct use of these techniques, a series of biological and biochemical validations are necessary to ensure that the metabolites analyzed first extracted from our samples are a true reflection of the biological changes in the animal. We can see in the screen, we can carry out a biological validation, physiological validation, by demonstrating a cause-effect relationship between the stimulation of the hypothalamic-pituitary-adrenal-gonadal axis and the corresponding increase in the concentration of steroid metabolites found in secretion samples. We can see in the screen that an example of this physiological validation. We made this validation with alba-tacaraya, injecting ACTH, and watching after 24 hours the excretion of the peak of excretion of the cortisol metabolites. A biochemical validation is also carried out for the correct development of these assays by demonstrating the immunoassay specificity, sensitivity, and precision through parallelism tests in order to determine at what dilution the hormonal extract obtained from our sample should be assayed to achieve at least a 50% binding with the antibody. An accuracy test is carried out to determine to what extent the matrix and debris of each sample affect the antibody hormone binding in the immunoassay. Using these techniques, we are able to evaluate both in the wild and in captivity aspects as diverse of the animal behavior as impact of hierarchical positions, cooperative bonds, predator presence, a study of reproductive cycles, seasonal variations, sexual and behavioral differences associated with hormones, effect of environmental toxins on the endocrine function, stress, and even more impact of the human activities on the animal welfare. Making advances in the field of endocrinology is a necessary complement in behavioral studies in wild animals and zoo animals in order to prioritize the well-being and health of them in support of the welfare, protection, and conservation of species. Thank you all for your attention.